Good morning, and welcome to the second ATCO SpaceLab Speaker Series event of 2024. My name is Alicia Tropak. I am a member of ATCO's transformation team, and I am pleased to be your moderator today. As a settler, I acknowledge the Indigenous peoples are the traditional custodians of the lands that we are on today. Since this is a virtual platform, I would like to recognize all of the various traditional, ancestral, and unceded lands on which we do our business. I do this to reaffirm my commitment and responsibility in improving relationships between nations and improving my own understanding of local Indigenous peoples' histories, languages, and cultures. I am honored to live and work in Treaty 7, the traditional territories of the Blackfoot Confederacy, Nitsit'tabi, comprised of the Siksika, Kainai, Piikani nations, the Tsuut'ina Nation, and the Stoney Nakoda, including the Chiniki, Bearspaw, and Good Stoney First Nations. The city of Calgary, Mohkinstsis, is also home to the Métis Nation of Alberta, Region 3. Our speaker today is joining us from Treaty 6 territory, a traditional gathering place for the Dene, Cree, Blackfoot, Dakota Sioux, Saulteaux, the Métis Nation of Alberta, Region 4, and many other histories, languages, and cultures that influence that community. Now a quick comment on the lecture format. Today's lecture is a one-way video and audio format. Please ask questions by using the question icon on the right of your screen. We will open the question functionality shortly after starting today's lecture. I encourage those of you, like me, who may be new to today's topic, to really embrace this opportunity to be curious, ask questions, and learn something new. I would now like to hand it over to my friend and colleague, Andrea Kleiber-Langen, to introduce our speaker today.
Thanks, Alicia. We're so pleased to have all of you and Dr. Thomas Hillen join us today. Dr. Hillen is an accomplished professor with the Department of Mathematics and Statistical Sciences at the University of Alberta, the Academic Director of Research, and the Site Director for the Pacific Institute for the Mathematical Sciences. His main research interests include the mathematical modeling of cancer, cancer growth, diagnosis, and treatment, with an emphasis on cell movement behavior. He applies movement models to a much wider range of fields beyond cancer, such as chemotaxis, wolf movement, sea turtle orientation, and tulip patterns. He has published over 100 journal papers in international journals and written 7 books. Dr. Hillen's areas of research include mathematical biology and partial differential equations, and he will touch on these topics today in a lecture on striped tulips and the mathematics of patterns. What a wonderful topic to kick off the spring season. We are honored to learn from you today, Dr. Hillen. The floor is yours.
Yeah, thank you very much, Alicia and Andrea, for this wonderful introduction. It's a great pleasure to be here and talk about one of my favorite research projects about striped tulips. Now, you might have heard in the introduction that I'm a mathematician, and you might be a little bit concerned that I try to kill you with mathematical equations. So I promise that is not my intention. And instead, this is a story I'm going to tell you will touch on many different areas and disciplines. So I will talk about history, I will talk about financial markets, I will talk about arts and poetry, but I will also, of course, talk about science, modern science, and and computer science, plant biology, and some detailed stuff, of course, as well. But then taking this story together, we will be able to solve a 350-year-old mystery right here at this talk. So let's jump in. First, let me introduce my collaborators. Aidan Wong is a undergrad student who just finished his undergraduate program, and he will enter a master program in Toronto. And then Gustavo Carrero is a colleague at Athabasca University here in Alberta. So what's the problem? Well, a problem, not the problem. The topic. The topic are striped tulips. So we all know tulips, and we all like tulips, and usually they come in bright colors of red or yellow or white. But then every now and then you see striped tulips. Nowadays, this is not such a big deal because you can just buy striped tulips in a store. But like 300 years ago, it was a big deal because the striped tulip variety was very rare. And not many tulips would show stripes like here. So they were brought to Holland in the 16th century, at the end of the 16th century, by botanist Carolus Clusius. And he planted these beautiful tulips, the striped and the non-striped, in his garden. And he got them stolen all the time because they were so beautiful. So at some point, he just gave up and he just sold all his tulip bulbs and said, okay, do whatever you want with them. I can't keep watch of all my tulips. And this initiated then the growth of this tulip as a market, and people would start to buy tulip bulbs. Of course, they're beautiful. But then this led to a real financial market where people would speculate on the price of the tulips and drive the price very high. So let me just see what do I have here on the next slide. Also, in those days, they didn't have cell phones, so they couldn't just take pictures or selfies of these beautiful tulips. So they hired painters. And then here you see two paintings, one from Morel and one from Bollinger from this time, where they painted bouquets. And we assume nowadays that this is a realistic depiction of the tulips, because at those times they were trying to paint quite realistically. And you see these beautiful tulips with the red and white stripes. And they can appear in different forms and different shapes, for sure. For this reason, the tulips are also called Rembrandt tulips. However, Rembrandt never painted those. Actually, he painted exactly one, kind of by accident. This is here. This is a picture of his wife, and she has a flower bouquet on her head, and there's one broken tulip visible there with the stripes. But he never really sat down to paint a bouquet of flowers. They're still called Rembrandt tulips just because it was a time of Rembrandt where the Dutch painters would paint those images. Okay, so let's talk more about this, the price of the tulips. So then merchants would speculate on the price of the tulip bulb for the next season. And then in October, November, they will put down option prices so that they can get the new tulip bulbs when they come out in the spring. And this went completely out of hand. So they paid about 3,000 guilders for a tulip bulb option, which would be the value of about 300,000 Canadian dollars nowadays. So a small house in Edmonton and Calgary, or maybe a garage in Vancouver, something like this. And this picture here is wonderful. So you see the merchant sitting on the table, and he has the tulip bulbs, and is selling it to a fool who's standing there and paying for it in gold. And then in, I think, 1637, I have it on the next slide, 1636, this financial bubble burst. And this is reported to be the first financial bubble which ever burst. On the left, you see a picture from the Amsterdam Tulip Museum, where the price of the tulip bulb was recorded from 1634 to 1637, so 3 years. And you see in particular in 1637 how the price really rose very dramatically. And then all of a sudden it collapsed. And nobody was interested to pay half a house or something for these bulbs. Thomson in 2007, you see here on the right, he revisited this story with the prices, and he found out that the really high prices were never realized. So these were option prices, but they were never realized and they were never used to buy tulips. Still, the price rose several magnitudes, more than doubled, and then broke down all of a sudden. Yeah. Here's a nice paper of my collaborator, Gustavo Carrero, about this financial bubble and how it arose and how it decayed. Okay. Now, here's a wonderful poem of Deborah Leipziger from 2013, so relatively recent. For decades, no one knew what caused the flaring, the feathering of tulips. Parrot-like, red on orange, peppermint red on white, black on tangerine, the eruption into flame. For broken tulips like Absamol or Mabel, what causes the tulips to break? The mosaic virus carried by aphids infects bulbs, and the flower breaks. Its hold on one color, the primary color suppressed and the lighter colors bleeding through. The beauty of a curse. Okay, so this is lots of facts hidden in a poem. So we need to look at this maybe a little bit closer. In particular, if you can see my mouse, the virus, right? It mentions here on the right, there's a virus involved. And this is, in fact, the case. It turns out, and this was shown in about 1930, that the striped tulip varieties are infected by a virus. And this is, when I read about this the first time, I found this super curious. First of all, I never thought plants have viruses. But why not, right? I mean, every species has viruses, so why should plants not have viruses? But then also, a virus is considered to be a curse, right? It's an infection. It's coronavirus. It's influenza, whatever. And here it leads to exceptionally beauty. It leads to these wonderful stripes. So apparently carried by aphids. So aphids carry the virus, bring it to the plants. Then the plant is infected, and then somehow, whatever magic, it makes these stripes. Yeah, so this is a very cool problem. And let's dig into this a bit more. So broken tulips are those with the stripes. They are weaker, they are smaller, and they are less likely to reproduce. And it was observed that it's transmissible from one tulip to the next. So again, this, of course, indicates a virus infection. And then Cayley and McKay in 1926, they found the virus, and they call it the tulip breaking virus. They found that this is very similar to another virus, which was already known for about 30 years or so, which is called the tobacco mosaic virus. So the tobacco mosaic virus is a problem for tobacco planters because it can infect tobacco plants, and then they don't grow so well. And they also show pigmentation patterns which do not look nice if they are made into a cigar. So this was a problem for the tobacco planters. Now, the tulip breaking virus is similar. It is a rod-shaped virus, has a similar size, and it belongs to the same family of viruses, which are called protivirus family. Okay, so the tulip breaking virus interferes with the pigments, and the pigments make the color, and they are called anthocyanins right here. So this is the color pigment we are interested in, which in the end makes the red stripes on white or the yellow stripes on red. So there are different varieties of anthocyanins, and I will talk about this in a second. So here's an interesting book by Karin Mölling. It's just called Viruses, More Friends Than Foes. And I read this at the beginning of the corona pandemic because I just wanted to learn more about viruses. And she gives a wonderful introduction to viruses and virus biology, and also lots of examples where viruses are very important. For example, they are part of your microbiome in your guts. So they are bacteria and also viruses, and they help you digest your food, for example. Or viruses are very important during pregnancy to establish the early fetus and the early embryo, and so on. So they also have very nice and very useful functions. But of course, they're also infectious. There's Ebola virus and others. So then on page 223 and 224, she wrote the following thing. So she described the stripes in the tulips, and she also describes that a virus is made responsible for this. And then she writes, Alan Turing described the mathematics leading to stripes. An activator and a long-range inhibitor are interacting, but I do not know which of the two is a virus. So what's going on here? Well, she mentioned Alan Turing, and I have like my next 3 or 4 slides on Alan Turing. So Alan Turing is known for many things. He's really a genius in the beginning of the 20th century. And one thing is pattern formation. So the Turing model has been used to model seashell patterns or animal patterns or fish. Fish are also animal, fish patterns. And so Karin Möllin then made the connection that the Turing mechanism might also be able to explain the striped patterns of the tulips. And then Turing's mechanism then involves 2 things, an activator and an inhibitor, which have to interact in the right way to then make the patterns. And when I read this in her book, I thought, I can do this, right? When I learned mathematics, I learned about Turing patterns. Actually, when I teach in my classes, I teach Turing patterns. So I know the technology which is behind this. And I thought, I can do this. I can find the patterns and I can answer Karin Möllin's question, who is or what is the virus? Is the virus the activator or the inhibitor? Okay, so that's the real story I want to tell you. And let me first talk about Alan Turing. Alan Turing lived from 1912 to 1954. He was a multi-talented mathematician, and nowadays several things carry his name. So here are 3. So first of all, Turing decoded the Enigma. Enigma was a coding machine from the Germans in the Second World War. Right, here's an Enigma shown. And you all might have seen The Imitation Game, this wonderful movie with Cumberbatch, who plays Alan Turing, and then builds this great machine, which is seen behind him, which then he then uses to uncover the code or to break the code of the Germans. So this is the same Turing. It is a wonderful thing. He also invented a Turing machine, only theoretically. So he didn't build a Turing machine, but he thought of the main principles, how a computer should work. And then the first computers, they use these hole punch things, right? And so they're built from the recipe of Turing, based on the Turing machine. And then, of course, nowadays every computer uses these ideas. Another thing which carries Turing's name is the Turing test, and this is cool too. Here the question is if a computer can ever be as good as a person, or if a computer can ever impersonate a person. And then the Turing test is as follows. You have a tester who would write chat messages into your computer, and then either a human answers or a machine answers. And the tester needs to decide, is the answer from a human or is the answer from a machine? And in the early 2000s, this was a hot thing. And for example, these chess computers, what was it, Blue something, I forgot. Anyways, these chess computers would participate in these tests and many other things. Well, nowadays, though, we have ChatGPT. So I think this problem is solved. I doubt that a tester can decide between ChatGPT or a human. But maybe they could. Yeah, I don't know. I don't know what's going to happen with the Turing test. But this was done on a yearly basis, machine against human, and to see who is better. So very interesting. So the Turing stuff we are interested in are these patterns. So here we have a fish. And then here on the left, we have a pattern which is computed, actually here on the bottom. That's a pattern computed with the computer based on Turing's model of an activator inhibitor. And you see they appear similar. So it's on a finer scale here in the picture than in the fish, but it also has these labyrinthian folding kind of patterns happening. Okay, so zebra patterns here, there are very different kind of animal patterns. There's a giraffe in there, there's a leopard in there. There are seashell patterns. So all these patterns have been described by Turing's mechanism. And so now we can understand why Karin Mölling thought about this when seeing the tulips. So there might be something here. Let's look into this. So now I got you the first mathematical formula. Don't be scared. Of course, I have to show some math formulas. Also, I'm sure there are some nerds in the audience who would appreciate this. But even if you're not such a nerd, let's see if we can make this work. So to get these patterns, the Turing patterns, we need two morphogens, and I call them U and V. Later, it will be the virus and the anthocyanin. Okay, it will be the pigment and the virus, but in general, they're called U and V, and they satisfy a partial differential equation where you have a right-hand side F and G, which describes the interaction. So it describes how the virus inhibits the gene expression for the pigment, and on the other hand, the pigment kind of inhibits the virus infection and how they interact. And now a common technique, if you are studying such an equation, is linearization, and then you look at eigenvalues. Okay, eigenvalues of the Jacobian matrix, which we call DF. And the important thing is the sign of the Jacobian matrix of this term FG. Right, so again, this is more like for the experts in the room, but then there are two sign patterns which are possible, plus minus, or minus minus, plus plus. And both of these lead to pattern formation. Other signs will not lead to patterns, okay? These are the two possible ways. On the right is called the activator-inhibitor mechanism. This is what Dr. Mölling was talking about. On the right is the substrate activator mechanism. This is what Dr. Mölling was not talking about, but this is what the virus is using. So in our analysis, it turns out we have a substrate activator situation and not an activator inhibitor situation. But anyways, it's very related mechanisms. Okay. So the goal is build a simple mathematical model. Well, simple is always relative. So build a mathematical model which captures the essence of the formation of the patterns. And then to answer Dr. Mölling's question, is the virus the inhibitor or the activator? So I go back to the previous. So here I said we have at least two ingredients. It turns out we need three. So here are the main players for the modeling. On the left, we have anthocyanin. And now you see different anthocyanins have different colors. And they also appear in vegetables and in fruits and in plants. And so we are mostly interested in the red or a purplish red color for the tulips. But different anthocyanins carry different colors. And then in the middle, we have proteins. Proteins are inside the cells of the tulip, and they are used to build virus. And here is a virus, a rod-shaped virus. And you see these yellow extensions here. These are all proteins, or the green balls here. These are all proteins. So we take the proteins and we build them together in this virus. So the proteins build the virus. Okay. And so now we need to try to understand the interaction between these three main players: the pigment, anthocyanin, the protein, and the virus. And I think this leads us then again to some equations. Yeah. Okay, so here's the anthocyanin which we are looking at. Here's a chemical formula for it. And the tulip-breaking virus interferes with the expression of anthocyanin. And the broken sectors represent virus-infected tissue, where the normal areas are virus-free tissue. Absence of color is due to the absence of anthocyanin. Okay? And also the other way around, anthocyanin inhibits virus expression. So this has been also reported for the tobacco mosaic virus, TMV. It has not been reported for the tulip-breaking virus, but since the tobacco virus is so similar, so close, we use the same mechanism here. So then this describes the anthocyanin dynamics. And let me show the picture. I think the picture is better than the formula here. So we describe the pigment with the big T. And when the pigment concentration is large, we see red. And if it's low, we see white. And the right-hand side here, this is a well-known model. It's called the Volperts model for gene activation. So this is used if somebody talks about how a gene is activated inside a cell. And it depends on the input signal row 0. If the input signal is large, this is the right picture here, then we have a steady state here on the right. And this means full pigment expression. So here the right one would be the yellow, the red case, full pigment expression. If the input row 0 is small, we get to this picture where we have a steady state here on the left, and this would be no pigment expression. So what then happens later is that we flip between the left picture and the right picture, depending if we are on a pattern stripe or no pattern striped. All right. I mentioned the input is important. So the input is here the red thing. And now we make this dependent on the virus. So the virus is messing around with the input to the pigmentation. All right. And then I need something for the virus, which is the equation in red here, and something for the proteins, which we call building blocks here. So I spare you the details, okay? What this does, as I said, it balances the interaction, how the protein is used to build virus, how the virus interferes with the anthocyanin, and how the anthocyanin interferes with the virus. All this is based on biology, on biological papers, which we read, and so on. So then this is a whole bunch of terrible math equations. But then there are always students who like this challenge and who like to look at this. And so we were glad to find Aidan, who then could work on these equations. Okay. So there are lots of parameters. There's a row 0, there's a little r, there's a delta, there's a row t, whatever they mean, they need numbers. So the first thing we had to do is we had to find all these numbers from the literature, and here they are. I mean, you can imagine if you have to do this, this takes months, right? I mean, I can show this like in a 3-second slide, but of course, this is months of work to figure out all these values. So this is not to be underestimated. Okay, so here's the equation again. All right, so I have to do one more mathematical detail, but then that's probably it. It turns out if we do the parameter values for rho, for r, for delta, for rho t, for rho b, for sigma, whatever, we find that the values in the first equation are all small, and the values in the right equation and the other two equations is all large. And this is a great observation, because then we can use an epsilon to indicate, hey, these are all small terms, but these are all bigger terms. And this will split up the equation into 2 bits, into the small terms and the big terms. And there's a mathematical theory which is called multiscale analysis, which then allows us to look at the big terms separately and the small terms separately, and then piece it together. And this is really a great key to analyze this further. So let's look at the big terms, the large terms. This is called the fast system right here. So it's the same as before, except the t equation. The t equation I throw away. Okay, so then this one I can do exactly the Turing type analysis. I can do linearization. I can look at the sign pattern. And remember, there were 2, right? There was the plus minus, or the minus plus plus. So this is where we get minus plus plus. And this is great. This put a big smile on our face, because this is the activator substrate model, which was studied by Gira Meinhardt and also known as the Schnakenberg model, studied by Schnakenberg. Okay, but anyway, so it's one of the cases where we can expect patterns. Awesome. All right, so then we have, what do we have? B is a long-range substrate. These are the proteins. And V is a short-range activator. This is a virus. So now we answer Dr. Merling's question. So now we see V is an activator. The virus is actually the activator in this pattern forming mechanism. Great. Okay, so let's do this in a computer. We know all these values for the coefficients. We can solve this numerically. And you see the x direction here from 0 to 10. Time goes in this direction. And you see the formation of patterns. So these are what we call patterns. It goes up and down and up and down. So there are patterns for B and for V. Okay? So you could say this is kind of the tulip stripes. Or you could say, well, not really. They don't look like tulip stripes. I have never seen a blue-yellow tulip. Okay, so we are maybe not quite finished. Well, one thing is, we need to put this now together with the anthocyanin dynamics, which was the first equation, the T equation, right? I said that the coefficients are small. I throw this away. But now I take it back. I need to use it. And then I have all three models, and I get the same patterns. So what do we see here on the right? We see in red is the virus concentration going up and down. In green is the pigment going up and down, and in blue is the building blocks, the proteins. They're all oscillating with the same frequency. Yeah, and then you can call this a tulip pattern. So does this look like this? The two pictures? Huh. Yeah, well, with a little bit of fantasy, maybe. So it's a good step, but we are not done. So we found a mechanism, this substrate activator mechanism, which makes patterns, but this is still not a tulip pattern. What is missing? Well, missing is that the tulip petals are growing, right? The tulip petal is not a square box or anything. It is growing. And it's growing over time, and at the same time as it grows, it lays down the pattern. So here are petal shapes. These are petal shapes, and you see different colorations as well, but they don't have stripes. But you can see nicely here, or actually in any of these, they start growing at the base, and then they grow out like a fan. Okay, they become wider, and then they become smaller on top. So have maybe a round shape here, or maybe a little bit more pointed shape here, or they can have other shapes as well. I mean, there are so many different tulip varieties. But what you also should see a little bit is the veins. So the veins fan out from the middle and they spread out left or right. I'll show you a better picture later. And then these veins, they can, of course, transport nutrients, and they can also transport virus particles and so on. So we want to use the above pattern formation mechanism, but now put it onto a growing tulip petal. All right. Yeah, growing domain. And there we use a theory of a friend of mine, Edmund Crampin, who unfortunately passed away way too young at the beginning of the COVID pandemic. But nevertheless, he developed methods to study these reaction diffusion equations on growing domains. And so it's a great opportunity to use his work for us. So what we do is we have a growing domain here, the 0 to LT. This should describe how the tulip grows. And I'll show you what this L is later. So this describes the form of the tulip petal. And we do some rescaling, and then we get a new differential equation, which includes the L in many places. Here's the L square. Here's the time derivative L dot. Here is L itself, etc. Okay, so just to honor Crampin, I show this here. So this is what we have to do with our model. In particular, we need to know what L is. What is the shape? What is the shape function? So we went to the literature to find tulip shape functions. And of course, it doesn't exist. Nobody has really measured how a tulip petal grows. But people have done it for roses. So we started with the rose petal function, and it is here. It's called the rose curve function, and has a wonderful formula. So it's rho times the square root of eta t, square root of 16t plus 19 eta minus 2t squared minus 3 eta t plus 1. I like this. I mean, whoever comes up with such a formula? I certainly didn't. Okay, but if you plot this, if you plot this, you do get a shape which kind of looks like a rose petal, maybe a little bit. Right, so here we plotted this this rose shape. Time goes from top to bottom, from 0 to what is this, 25. Okay, and then we overlaid our mathematical model on this growing rose petal domain, and we do get stripes, right? We do get stripes, white and red for the pigments. For the proteins, we illustrate this in green, and for the virus, we illustrate this in red. So somehow it seems to work, but for roses, and then roses are not tulips, and I've never seen striped roses before. So, so I think this is not the right thing to do. So we need to go back to the tulips. And so what we then did is we took tulip petals and we put them on a light source so we can shine light through them. And we took a picture with our cell phones and blew up the picture and make it really big so that we can see all the veins very clearly here. And then Aidan sat down and he measured arcs from the middle to the side, which are perpendicular to the veins. And the idea was that cells along such an arc have the same age. They have the same distance to the origin here, and they are perpendicular to the veins. So these are cells of the same age, which means these are cells which have participated in the pattern formation process together. So this is our idea how to parameterize the shape of this tulip petal. And then we need two things. We need the length of this arc, or the double length of this arc, and we need something like a radius curvature information. So how much is this bent over? And we can do this. So there are two ways. There are ways to measure the curvature R and the arc length along this curve L. So Aidan sat down and he did this very meticulously and very detailed. And then here are his results. So the arc length is the length of this perpendicular line which he drew, and these are his measurements. And in blue we overlay a function which can describe it. And then the radius of curvature is this. So it's first high, then low, and then it becomes higher again. And from the error bars, you see that the measurements are rather detailed. So he did a really good job there. Okay, so now we have the measurements and we want to fit a curve through this. So for L, we use a logistic curve. And for the radius, we use a Volpert curve. There are different ways you can do this. And then you can write it down as some formula. So logistic curve, the blue curve is this. And the R, the radius curve is this. And you see they fit through the data really, really well. All right, so how do we put this together then? I said earlier in cramping, in the growing domain thing, we need to know what the L is. Now we know what the L is. L is this, okay? So we can just put it into the equation and solve them and see what we get. And this happens in the next slide. First of all, right, blue is a straight domain and green is a transformed domain. All right, so here for each time step from 0, what is it, 1/2, 1, 1.5, 2, and so on, I first of all plot the length of the domain in a blue line. Okay, so this blue thing is the length of the domain from the previous data. Go back from the arc length on the left. I plot this as a blue line, and then I use the curvature radius to bend it over so that I get the green line, which is now curved. Okay, so the blue line at 12 is then bent over to the green line, which is touching at 12, which then gives me the tulip petal shape. And I do this for 10 and for 8 and for everything. I curve it according to the radius of curvature given here on the right. Okay, so this is a little bit tricky, of course, but then Aidan could figure this out really nicely. All right. So now we have a growing domain, which is blue. We can solve our equation on the blue and get our stripes. And then we bend it over into the green to make it into the shape. Okay, let's see how this works. So here is the growing domain length, and then this would be patterns now for realistic parameter values for our tulip situation. Here's the pigment, here is the substrate, here is the virus. And now we bend it into the green curves like I've shown before, and we get these. And now these look absolutely fantastic. Right, this has the right shape. This is a tulip petal shape, much idealized, okay, but it's done on a computer. So, and it shows these beautiful stripes, and also how they're generated, and then they're split up into more stripes and so on. Okay, so this is a process. All right, so then we, if we change parameters, for example, the initial length, L0 is the length at the base of the tulip petal. And we have tulips of different sizes, of course. So if we go from small, which is 1 to 1.5 to 2, so this initial distance here, then we get more or less stripes. So the larger thing has space for more stripes, and the smaller thing has not so much space. Okay, but then these are the stripes we get. Okay, so let's compare this to real virus-infected tulips. So here on the right are again two pictures from the 17th century, where the tulips have been drawn and the stripes have been drawn, and then so the Camelot Van Vina pattern is matched really well by our model. It comes out really, really nicely. Well, that's a computer simulation. This is the historical tulip. Also the Vite Merveille, if we change the parameter values, we can also get patterns which are like more white on the outside and have the pattern more inside. And this corresponds also nicely to these kind of tulips here. Yeah, so I think we're very pleased. So this works really well. Also, the growing domain really makes a difference. So now we can put it together really nicely. Right. So this is the story. Okay. So historically, people knew that the virus is involved, but the mechanism, how the virus leads to the tulips, was unknown. And this detailed mathematical description can actually solve this and can find exact conditions under which you get stripes. And you can also calculate how many stripes and so on. So it's a combination of the Turing type substrate activator system and a Wolpert gradient sensing mechanism. These are both well-known models, but we put them together in a new way. We estimated realistic parameters from the literature as much as we could. And we can answer Merlin's question. The virus is the activator. So I cannot wait for our paper to finish. So we're writing this in a paper. And I cannot wait to send it to Dr. Merling and see what she thinks. Okay. Where's my thing? Yeah, the Crampon theory of growing domains is essential here in this analysis. And our parameterization of a growing tulip petal is actually a new thing in itself. So tulip petals have never been parameterized in this way. And I think this can be useful for other things too. All right. Nowadays, however, striped tulips don't have virus. They are engineered. They are bred. You can buy them freely in the supermarket, and you don't need to be concerned about a virus infection. These are healthy tulips, and they're just genetically engineered to have stripes. Okay. So then I come to the end of my talk. So now this one, I took the tulip patterns which we computed, and I printed them on white paper. And then I bought a tulip from the dollar store, and I cut off the top, but I kept the bottom. And then I arranged the paper tulip patterns in this form, so to get a nice looking artificial tulip. So I thank you very much for your attention.
Thank you so much, Dr. Hillen. It's now time to begin our Q&A session. A reminder to our online audience to use the Q&A function on the platform. Our first question is actually from our event producer, Dr. Hillen. So now that we understand how the virus leads to the patterns, can we generate new patterns in nature?
Yeah, this would be nice, right? If somebody comes and says, for my wedding, I want tulip stripes in purple and gold or something. Yeah, no, this is not possible at the moment. We can understand the patterns which nature produces, but to genetically engineer the patterns based on anthocyanin expression in a healthy tulip is so far out of reach. Yeah, so we will not be able to do that.
Great. So we have a question from one of our online viewers, Patrick, and he's asked, was the mapping of the tulip arcs done by hand or with computer assistance? And how many tulips did you end up measuring?
Yeah, so this was done by hand, and we measured one tulip petal. But we measured it several times, so maybe, I don't know, maybe that counts as a repetition, but we use this one image which I've shown you.
Our next question comes from Alita. And Alita asks, is there a correlation between the shapes of tulip petals and the distribution of stripes?
Absolutely, yes. So if we, for example, use petal shapes which are more pointy at the top, then we would also get a different number of stripes at the top at that region. So it essentially depends on the radial distance along the tulip petal.
Thank you. Our next question comes from Scott. And Scott has written here, seeing as the striped tulips that you can buy are genetically engineered, I'm wondering how much control growers have as to what the final product looks like. Is it pretty random?
So that's a good question as well. This is not random, because in this breeding process, you typically keep the striped patterns you like, and you throw everything else away. So then these genetically engineered, they're all clones, so they will all show the same patterns.
Yeah. Great. Our next question is from Robert, and he asks, thank you for the fascinating talk. Are color patterns in vegetables like radicchio, I hope I'm saying that correctly, caused by a similar mechanism? Are viruses also involved there?
So there have been reported spotted patterns in certain vegetables related to anthocyanin expression. So this is entirely possible. But also it could just be, for example, potatoes. If potatoes get a bit older, they get black spots inside, which is then a completely different mechanism. But definitely they play, anthocyanins play a role in vegetable patterning for sure.
Thank you. We have a question here from Stalin. Stalin asks, could you give or provide a small background about how Turing patterns were formed to determine patterns found in nature? And quite an interesting way to use concepts. He said, quite an interesting way to use concepts in math to find patterns in nature. Amazing presentation.
Oh, thank you very much. Yeah. So you're asking for an intuitive way of Turing mechanism. So we need a short-range activator and a long-range inhibitor. This is many people have thought about this, but it's not so easy to come up with a good answer here. So in the case of pigments, so what is the virus is the okay, I'm sorry, give me a second. Need to gather my thoughts. So the virus is a short-range activator, which means the virus acts very locally and then destroys everything where it is. Whereas the cell produces proteins because it needs proteins for its normal function, and the proteins are also communicated to other cells, and they are the long-range substrate. So these two, this one local mechanism and the global mechanism working together, they then can lead to the pattern formation. So this is not a very good answer, I know. But yeah, okay. So you have to live with this. So, but you can look up Turing patterns on Google, right? And you find other explanations there.
Thank you. I think we have time for a couple more questions. At the beginning of your lecture, Dr. Hillen, you shared your inspiration for your research. How often are you and your collaborators inspired by historic events, art, or nature in your research? And can you share any other examples that you might be working on?
So yeah, this indeed happens very often, I think, so more often than I can deal with. So another example is I play piano and jazz piano, and as a jazz pianist, you learn about the modes. So I'm sure some of you have heard about modes and modal improvisations and so on. And also being a mathematician, I immediately saw a pattern. And then I thought, okay, I need to analyze the pattern of the modal scales. And so I did. And then this leads to other research. Oh, what is more relevant, let me give you one more example, is on cancer. Right, initially, Andrea said I work a lot on cancer modeling, which is true. And I read a paper about cancer microtubes. And so what that is, sorry, I need a minute or so. So glioma is, of course, a very aggressive brain tumor, which grows in the brain. And the question has been, how does it grow? How does it actually grow? And it turns out that these cancer cells send out very thin protrusions, like needles, to test the environment, and then they can grow along these needles, and they form a network where different glioma cells are connected by these microtubes. So in effect, it is a little bit as if the brain cancer tries to form another brain inside the healthy brain. And this is an absolutely fascinating story. So I thought then, okay, we need to understand this better and maybe come up with mathematical models for this as well.
Very interesting. No, we have two more questions for you before we can sign off. Patrick asks, what's the mechanism behind the viral degradation that appears in your equations?
So how the viral degradation is just a normal decay. So it would essentially be a washout. I've shown you the veins along the tulip petal, which transports nutrient and water and so on, and also virus particles, and they would just be washed out. They would just be lost like this. Yeah, I think that's the main contribution to that term.
Thank you. Actually, if we have time, Dr. Hillen, we've got a couple more questions as well that are just coming through. So that's wonderful. Thank you. And please keep sending them. Our question from Robert is, do the L0 initial length parameters you used match an actual tulip petal?
And yes, it does. It's in a range of 1 centimeter, which is the base of a normal tulip petal. Yes.
Wonderful. Brian asks, is there an error term in those equations? Or is there a time invariance with the parameters with the stochastic equations? As in, given the same starting patterns, can the tulips grow to have different patterns?
This is a very good question. I love it. So if you look closely at the patterns I've shown you, they are super regular. So first of all, we reproduce them 100% of the time. Same initial condition gives the same pattern. And then they are super regular. And in nature, they are never that regular, right? So in nature, you have disturbances, you have variations in temperature, you have maybe even birds nibbling on the tulip a little bit or whatever. So these stochastic random effects are not included in our model. And I think if we include them, this would make it even more realistic.
Wonderful. And our final question for today comes from Scott. And he asks, To expand on a previous question, I was wondering who were some of the first people to determine that Turing's work could be applied to understand things that we observe in nature? Or what were some of the first use cases?
This is a wonderful question as well. I love it. So I love it because it was also a discussion in the scientific community. So I think the first conclusive proof that Turing's mechanism leads to pattern was done in chemistry. It's called the CIMA reaction, CIMA, where different chemical ingredients react, and one diffuses fast and the other diffuses slow. And this was showing exactly the patterns which were predicted by Alan Turing. So I think the first conclusive situation was in chemistry and not in biology. And then Gira Meinhardt has done a lot on seashell patterns, which are also, I think, very conclusive how these patterns arise. And then many researchers, maybe starting with Jim Murray, looked at animal skin patterns like leopards or giraffes. And fish. Kevin Painter, he looked at zebrafish pigmentation patterns. And in that case, you can identify the pigment cells which are moving about and making the patterns. So now we have very good examples. But the first one was in chemistry.
Fascinating. Well, thank you, Dr. Hillen. That concludes our session for today.
Yeah, thank you very much, Alicia, and for all the questions I got. It's good.
Wonderful. We'd very much like to thank as well all of my colleagues who helped put together this speaker series event for us together today. To everyone that has joined us online, we very much appreciate your interest. I'd also like to remind you that this lecture, along with all of our past lectures, are available on our YouTube channel. Please stay safe and enjoy the rest of your day. Thank you for joining us.